Hello everyone, welcome to our first evening for our Soapbox event for our program called Remaking Life. This Soapbox series, we have 4 talks and they all explore different aspects of synthetic biology. On everyone's seat, you guys had one of these cards that outlines what each of the 4 talks are. They're going to be every Wednesday night from 6 to 7:30 in the museum right here. So hopefully we'll see you guys coming back for that. Just so you know, at the end of the night, we do have a small survey that we'd like you to fill out, and then a small raffle prize. So if you do fill it out, you get a chance to take a little gift from the museum home with you. So hopefully you guys will be willing to spend about 3 minutes to help us improve on our series. So our first speaker for tonight is John Dench. Is Christopher Voigt, who is the professor of biological engineering at MIT. If Chris would like to come up, and we'll hear a little bit about his research and how it fits into our title for tonight, From DNA to Designer Genomes. So I hope you guys enjoy. After Chris's and John's talks, we'll then have small group discussions and then an opportunity to ask questions as a larger group. And so hopefully you guys will enjoy this conversational setting as well.
Great. Well, I'm glad to be here. So I'm a professor in the biological engineering department at MIT, and I'm a synthetic biologist. And so what that means is that I aspire ultimately to be able to design entire genomes from the ground up. We want to understand how every piece of DNA works together, where if we have a living system that we want to build for any particular purpose, we can go in and figure out which pieces of DNA need to be put together in order to create that living system. And so in this short talk, I really want to do 2 things. Very quickly, I want to do just an overview of what synthetic biology is and where we stand now and where we're envisioning we would go. And then the second part of the talk, I'm going to describe some of the research that is out of my lab, where what we've done is created a programming language for bacteria. So this is a language where you write at a very high level what you want the bacteria to do, and then the computer figures out the DNA sequence in order to do it. So first, synthetic biology is a discipline that's really emerged over the last decade to try to get better at engineering biology. And so some of the principles of the field are to try to break down individual biological functions into the unit of DNA that encodes that function. And that's known as a part, a genetic part. And it's just a snippet of DNA. And then a big part of what the field tries to do is figure out how to put those parts together in order to create larger systems and ultimately whole organisms where you understand how those parts work together. And so what I'm showing here is that basic idea where you start with individual parts that then gets put into a DNA sequence, which on some level is just information. It's just the A, T, G, and C of DNA. And so you have that sequence, and at some point it's on the computer. And then that has to be converted into a physical piece of DNA. And I'm showing a picture of that there. This is a circular piece of DNA that then gets put into the cell. And inside the cell, it then boots up and runs whatever parts and whatever system has been put onto that physical piece of DNA. And so the field is really about trying to make each step of this easier so that you can better realize what it is that you need to build in order to get the desired function. So if this seems somewhat futuristic, you might be surprised that it's very much in play already. So this is out of a White House report from several years ago, looking at the current US economy from what they call genetically modified stuff. So this is everything that we currently get from engineering organisms. If you add it all up, it's $350 billion a year, which is about 2% of the US economy. It's about the same scale as global semiconductors. And so it's an enormous existing economy that goes from everything from making precursors to pharmaceuticals to materials. A lot of the Prius actually comes from genetically modified precursors to the food that we eat. Now, what I'm showing at the bottom are some of the actual chemicals that cells have been engineered to produce. And you don't have to be a chemist to recognize that those are actually quite simple structures. There are a few carbons, some oxygen, some hydrogen, but by and large, the same types of chemicals that we get out of the petrochemical industry. They're not nearly at the level of sophistication that you can get out of biology. And so this is an example of one of the types of molecules that biology can produce. This is an organism that lives in the ocean, and it uses this molecule in order to make you sick. And you see in comparison to the molecules that I was showing on the previous slide, which are very simple with a few carbons, this looks like a centipede of atoms. And getting this level of precision is really difficult by chemistry, but biology does it all the time. And it's not just chemicals, there's also materials that we could produce. These are the structures of diatoms that are living in the ocean. These are made out of silicon, a couple microns across. And you can see that these organisms are able to build very precise structures that have all sorts of applications in our world, from catalysts to photonic and optical properties, to just about anything that you can imagine. And it's not just making chemicals or materials, but of course the wonder of living systems is that they can move around and interpret their environment and produce things that are needed at different times. And so this is an example of a project from some time ago out of our lab, where the idea was to try to program a bacterium that could serve as an anti-cancer agent, where you would actually inject an engineered bacterium into the bloodstream. It's able to figure out where in the body a tumor exists and then deliver a therapeutic. And so this is where a bacterium has to be programmed to be able to recognize different spots in the body in order to identify the one that's required. So now there's a huge gap from the enormous economy that we have around all of genetically modified stuff that's based on very simple molecules, to those things that we know biology can do, and the level of sophistication in the products and the therapies that we would like to get out of biology. And so this is where synthetic biology tries to jump in. And the real things that are limiting us currently from going from these simple products to the more sophisticated ones are really threefold. The first is that it's technically very challenging to build large pieces of DNA that have many genes on them. So we're still largely only capable of building a piece of DNA with 10,000, 20,000 base pairs of DNA. But if you look at genomes and what's required in order to create these other products, it could be millions of base pairs of DNA. And somebody has to physically put that together, and it's really tricky. The other hard part is controlling when different genes are turned on. When you're building a more sophisticated product, you have to make sure that some genes are turned on, and sometimes in some locations and other ones aren't. And by coordinating that, that's how you build up a structure. And we're only at a very rudimentary level of understanding what types of systems cells use in order to tell which genes when they need to be turned on. And then finally, the design tools that are available are atrocious. Even in the most advanced genetic engineering projects that are out there, it still largely involves a designer that's sitting there and physically picking up pieces of DNA and putting them together on the computer in order to try to concatenate out and build up a system. When I was a graduate student about a decade ago, I used Microsoft Word, and I had this giant Microsoft Word file that had all my favorite pieces of DNA. You became a bit of a DNA whisperer of figuring out exactly what sequences need to come together in a separate Word file, and then the whole thing would crash and be a disaster. And you got really good at being able to look at DNA sequences and see within there what's actually going to happen. So unlike designing a car or an electronic chip, there just aren't the computer-aided design tools available for genetic engineering. So now what our lab tries to do is to address all of these different problems, to go from simple, small genetic systems to the big ones that can involve many different systems and sets of genes, and controlling when they're all turned on at different times. And so this is an example of a design where the idea is to have a bacterium that makes antibiotic spider silk cloth. And so if you just think of what's required in order to do that, you have to have genes from the spider and the bacterium. You have to have a whole system that the cell uses to export and spin those fibers into threads. You have to have all the genes to make the chemical that serves as the antibiotic. And then you have to tell all those different systems to turn on at different times in response to different conditions. And so to do that, you can link turning on those different systems to sensors in the cell, and then circuitry that then processes the information from those sensors to figure out what system needs to be turned on at what time. And so what I'm going to tell you about is one project in the lab on the left-hand side of this graph, where what we've done is created a language so that you can take any computational operation, anything that you want to put in a robot, for example, program it using a language that looks like you're programming a computer, and then it figures out the DNA sequence that puts that computational operation into the cell. And so this is what that looks like. So you start out on a computer, and in a high-level language, if you've ever programmed a computer or Lego Mindstorms or Scratch or anything like that, you're familiar with what a basic programming language looks like. And very much following that type of syntax, you write out the computational operation that you need for whatever application. And then the computer figures out how to take that file and convert it into a circuit diagram as though it was an electronic chip, figuring out what type of underlying computational functions you need in order to build that bigger circuit. And then in the next step, the computer figures out which pieces of DNA need to be stitched together in order to create that circuit so that it'll operate inside of a cell. And then that creates a DNA sequence that's the output of the computer program. And now you can email that off to a DNA synthesis company, and in a couple of days they'll send you back your piece of DNA that contains that encoded program. So if this all sounds very foreign, I have a slide to try to help you understand how you actually would encode the basic units of computation inside of a cell. So the way to look at this is that the line is a piece of DNA, and what I'm showing in the box is everything that's necessary for the cell to sense something. So this is behaving like a sensor, just like a Lego Mindstorms sensor or something that you would use in a robotic system to receive information. And so on that piece of DNA, the big arrows are genes, and they make proteins, which are the red boxes. And then the arrows that are bent are what are known as promoters. And that's where other proteins bind in order to turn on other genes. And so what happens in this sensor is that there's a signal, so that might be a chemical, and that's the small red circles. And that binds to the protein. And when it binds to that particular protein, it then recruits another one that then creates a flux of what's known as RNA polymerase, which would then turn on the next gene that this was connected to. So in the presence of signal, it turns on that next gene. And now, unlike an electronic circuit where the electron would be the way that we carry a signal, it's going to be this flux, this movement of RNA polymerase that's going to carry our computational signal. And so if you have a high level of signal that then turns on that flux, if you take that signal away, it turns it off, and you have a lower flux. And so you can black box, so you can forget about all the biochemistry, and just imagine that I have a piece of DNA, a snippet, and it's going to act like a sensor. And you can have it where it's just, if you have the signal present, you have a high flux out of that box. And then if the signal is absent, you have a low flux out of that box. And so that's what's going to then be how we can connect to that, to a circuit. Now, in our lab and in others, we've compiled a list of about 50 different sensors that all respond to just about anything that you can imagine. So we have sensors, which again are pieces of physical DNA that can respond to color. So we can make cells see red and blue and green. We can make them respond to touch or signals that are coming from UV. or toxins, or metals, or gas like oxygen. Just about anything that you can imagine, we have a sensor in the lab for that type of signal. And so this is what gives us this Lego Mindstorms-like kit, whereas we're imagining what we want the cell to do, we can pull out all these different sensors that respond to these different signals and have a way for the cell to be able to respond to the things that we need it to. So then once we have these basic sensors that turn on and off in response to a signal in a way that involves an RNA polymerase flux, we then have to apply a circuit to it in order to process that signal. And so this is fairly simple, where a circuit, before I tell you about the biochemistry, what it does is it takes one RNA polymerase flux as an input and then converts it to a second output. That's also an RNA polymerase flux. And when you do that, it's relatively easy then to connect these together, where you can take a sensor that converts a stimulus into a RNA polymerase flux, and then you can concatenate these circuits together where one feeds into the next, which feeds into the next, and so on, building up our signal processing that we need to do. And then the output is an actuator, which takes that signal and converts it back into a biological response. That's something that the biology would be doing. So once we did that, we had to figure out, well, what type of circuitry would we want to have in that black box that would allow us to build whatever we wanted to based on what a programmer would write in this language. And so to do that, we turned to some theory from computer science, which is surprisingly simple but very powerful. And that was looking at types of gates that are what are known as Boolean complete. And all that means is that there are special types of gates for any computational operation you can imagine. Anything you've done on a computer can be broken down to just that one type of gate. And if you have enough of those gates, you can build up anything that you want. It's very hard to wrap your mind around, but it's really the basis for digital computing. It's what allows us to do so much on a computer. One of these gates that's Boolean complete is what's known as a NOR gate. And a NOR gate is very simple, where it has two inputs and one output. And it's only on if both the inputs are off. And if either of those inputs are on, then the output is off. And so that's the logic operation and the symbol that's used for a NOR gate. Now, one of the things, if this is new to you, that I like to show, so a lot of people aren't familiar with this concept. And believe it or not, one of the ways that this was actually used was in the Apollo 11 missions to the moon, where all of the circuit boards in those missions were just based on this one circuit function. So if you took all the guidance systems and the emergency systems and everything that was in that shuttle, it came out to about 5,600 NOR gates that were all connected in order to perform the computation that was necessary to take that spacecraft to the moon. And so if we had 5,600 NOR gates that operated a bacterium, we could reproduce the Apollo 11 guidance systems within the cell. So in order to do that, we had to come up with some biochemistry that would then take that same logic function, but then have it be, instead of electrons, be these RNA polymerase fluxes that allow them to be connected. And as it turns out, it was a little easier to do this than we thought it would be. So this is how you encode a basic NOR function in a cell on a piece of DNA. And we're going to start with what's known as a NOT gate. And a NOT gate just does the opposite of whatever you tell it to do. And so if you look at the first bent arrow at the top, that's a promoter, and so that's driving RNA polymerase flux across the boundary, except now we have it turning on the green gene, which produces a protein that turns off the second promoter. So if the first one is on, and you have a high RNA polymerase flux, the second one is off, and you have a low RNA polymerase flux. And the function that that produces is shown at the bottom. And so you can then take this basic NOT gate and expand it very simply into a NOR gate by having a second promoter upstream from that. So now if we have two promoters on the DNA, if either of those promoters are on, then it's producing the repressor, and the third promoter is off. And so you can see that at the bottom, where only if both those first promoters are off is the third promoter on, and otherwise it's off. And that's the basic NOR function that we need in order to build up this, this computation. So we went through a lot of effort once we came up with this gate in order to identify a lot of gates that would work well together and that they could be reused on DNA without interfering with each other. And so at the end of that, we ended up with a library of these gates where these are all different, based on all different proteins that don't interfere with each other, where they can be connected together in order to build up more complex computation. And so the way this works is the following. On the top, we have a circuit diagram for a simple computational operation. And then we have to somehow use the gates to convert that to a linear DNA sequence that encodes that operation. And so that starts out where we take 3 of the NOR gates, we take the sensors, divide them up, and then selects those gates that connect well to each other, splices them together, and then that reproduces that logic function, but now on a linear piece of DNA, where instead of wires that carry an electronic signal, we have promoters that are then driving RNA polymerase splicing. So once we had that basic structure, I turned to a colleague in electrical engineering to figure out what the language is that we should use in order to capture whatever computational operations somebody might want to build. And to do this, we turned to a language called Verilog. It's very much like C, if you're familiar with that. It's been around since the 80s, and it's the way that electronic chips are programmed. And so we needed to hack this software so that instead of compiling to a silicon chip, it would compile to a piece of DNA. So this is what that software looks like. So it starts out where you put in a desired circuit function using this language, and then everything else is hidden to the user. So that when you hit compile, it then creates the circuit diagram that has all of the NOR gates connected in the way that creates that operation. It then selects from the database which of the gates will work well together, assembles it onto a piece of DNA, and the output is the DNA sequence. And so this is the way that the software works. It's going to be released on the web. We have an iPhone version of it. You sign in, you figure out what sensors you want. So you say, I want the cell to respond to other cells and oxygen, or I want it to respond to a toxin and blue light, whatever it is that you want that's in the sensor library. You then write the language that you want, how you want the cell to respond to those sensors, what that circuit function is. And then you connect it to the outputs. And this is controlling what the cell is doing. You select whether you want E. coli or human cells or other bacteria or whatever organism it is you're compiling the program for. And then you connect it to the output. In this case, these are just proteins that change the color of the cells. But you could also select to have it turn on pathways, to turn on different genes in the genome, basically connected so the output of that circuitry is having some biological function. You then go back, you validate and make sure that that code is correct, and then you run it. And once you run it, then it takes the syntax, it takes the sensors, it takes the things you want to control, and it puts it all together. It comes up with the circuit diagram, goes to the gate library, assigns gates at all the locations, comes up with the DNA sequence, and so on. So this is the output of the program. This is the circuit that it wrote, which genes are turned on in which conditions. And let's see. And so this is saying that all of the gates properly connect to each other like a puzzle, and that you would expect this to be functional. This is giving the experimentalist the prediction as to how it's going to perform so people can directly match it, and even tell you whether it's expected to be toxic or not when the cell carries them, whether the cells are going to die. And then this is the DNA sequence. And so this is where the computer went in and it figured out for this circuit function exactly what DNA sequence is needed. And we're going to have it when it's released where there's a button you push at the end. You hit that, this gets sent electronically to a DNA synthesis company, and in a couple of days you get a tube back in the mail or your kids get a tube bag in the mail that is the DNA sequence that they can then put into their cells. And so we've tested this on many different circuits, and it works very well. I won't go into the details because of time. But we've actually tested it on a megabase of DNA, showing that just about any circuit function that you could possibly want to build, it does so with a pretty high fidelity. We've also shown that you can then connect the output of these circuits to control different things. And this is just a few of those things. On the top, we have metabolic pathways, so telling the cells which chemicals to produce, in this case, changing the cell's color. In the middle, we have a function that's quite complicated. This is a nitrogen fixation. This is important in agriculture for converting atmospheric nitrogen to fertilizer. And we show that you can put that 16 gene pathway as an output to a circuit. And at the bottom is salmonella. This is the system that it uses to inject proteins into your cells to make you sick. And we've shown that you can put that under the control of one of these circuits. So we've started to link all of these different pieces together where you have sensors that you're using in the cell in order to receive information from the environment, circuitry to be able to program that cell to respond to the sensors and figure out what it should be doing when, and then the mechanisms whereby we can connect it to complicated multi-gene functions to then build up structures to enable the cells to do what we ultimately want them to. And so with that, I will thank my lab and take any questions.
So I actually save the questions for the end after we hear most speakers, but thank you very much, that was wonderful. So welcome everybody who wasn't here at the beginning. We're really glad you braved the rain to come see us tonight. This is our first of 4 soapbox series about synthetic biology called Remaking Life. Our second speaker for this evening is John Dench, who is an associate director of the Genetic Perturbation Platform at the Broad Institute. We're going to be pulling out a couple more chairs for the people in the back, and you can also squeeze in with a couple open seats. We do want you guys to talk to your neighbors after this point, so it would be nice for you to have some neighbors around. Perfect, but with that, I'll leave it to John.
Great, thank you very much. So I'm going to take a very different approach from Chris's talk, and instead of building things up, I'm going to talk about how we break things down in order to understand them. So we should call this like catabolic biology or something, but it's really called functional genomics. But first I want to start off by, saying it's the most important slide too. By they mean a lot of the people that I work with, especially Megan and Emma, who are also here today in the back, answering questions online, and I'm sure we'd be happy to answer questions about what this stuff is really like in person, as long as I'm out of earshot. So in biology, it's not a stretch to say that what the first question of biology in biology, not naturalism, but biology, was what is a gene? And really the first person to gain any insight into this was Gregor Mendel, who was working with pea plants. I mean, these are the canonical freshman year biology. You cross a yellow pea plant to a green pea plant, and what are the fraction of offspring with pink peas or pink peas, yellow peas or green peas? And he was the first person to say, well, heredity, this is something that allows you to predict the future, and that's a really powerful thing. I should point out that this XKCD cartoon is actually accurate. My wife and I, we did this on our second date, decided that yes, there's a chance we could have a blue-eyed child, and thus we continued dating. And we do have a blue-eyed child. So that was Gregor Mendel, working in the 1850s and 60s. The term genetics wasn't even coined until 1905, after Mendel was dead. His work basically went completely unappreciated in his own time. And the phrase gene to describe this discrete unit of heredity, that wasn't coined until 1909. So for the first half of the 20th century, the hunt was really on for, well, what is a gene made of? We know that there is information that is inherited from one generation to the next, but what form does that information take? And this is the canonical DNA double helix discovered by Watson and Crick in 1953, along with Rosalind Franklin, importantly. And they postulated, they say the end of this famous paper is not escaped our notice. The specific pairing we've postulated immediately suggests a possible copying mechanism for genetic material. So that's the idea that if you know the sequence of one strand of DNA, you know the other strand of DNA because they base pair together, and it's very basic code comprised of 4 nucleotides base pairing together. I should also point out that this was the first and last time that James Watson was ever understated about anything in his life. So then the question became in the kind of the second half of the 20th century, well, what are all the genes? So we know that information is comprised of DNA. Well, what's the human genome? So that's actually what led to what then became the Broad Institute, not long ago, the Human Genome Project. The idea that, well, we've got 3 billion bases, let's sequence them and find out what they all are. So that's where we were by the year 2001. The sequence of the human genome was completed. We had it, it was done. There was a library in England that, for God knows what reason, decided to print it out. So there's this like 80-volume set of books that's just nothing but bases. I don't think anyone's ever read it. But the point is, this doesn't make any sense, right? I mean, you can't look at this and say what the function is, right? I mean, this is the sequence of the human genome. This one, this one, this one, this. But this doesn't tell you function. It doesn't tell you what genes do. It just tells you what the sequence is. But we want to know function. So if you're given a DNA sequence, how do we determine a function? In other words, you know, we have the genotype. Okay, that's what the sequence of DNA is. That's your genotype. What we don't have is the phenotype. Okay, what that information actually does when it's in a cell. So if given a DNA sequence, because we have all our DNA sequence, given that, how do we determine a function? Well, the best way to determine a function in a complex system, or the simplest way to do it, is to break it. This is an experiment that Patriots have run for the last couple of years. For many years, when Rob Gronkowski was hurt, their offense was awful. But last year, he was healthy for the entire year, and they won the Super Bowl. And he's been healthy so far this year, which has been very good as well. So to find out how a system functions, you break a component and see what happens. So in this experiment that the Patriots have been running, Rob Gronkowski is good for the offense. But how do we do that in living systems? In other words, given a DNA sequence, how do we break genes? How do we somehow disrupt this flow of information so that we can break somewhere here and find out how it affects phenotype? So the way that we go and do this is a very new technology that starts with an interesting day in a yogurt factory, which if you Google or Bing, interesting day in a yogurt factory, you get no hits, because apparently these search engine things, this has never happened. But that is false. There has been at least one interesting day in the history of one yogurt factory. So this was a company that was making yogurt, and one of the many cultures you need to make yogurt is Escherichia. This is on the side of a Stonyfield yogurt can. This did not occur in the Stonyfield factory. So you have to grow up bacteria if you want to make yogurt. So they had happily growing Escherichia one day, but then viral contamination came along. That's bad. And they had a tube of mostly dead Escherichia. So that's bad for production, because now they can't make yogurt that day. But fortunately, this was a company that had some really good molecular biologists, and they decided, well, let's find out why this happened. So there were still some Escherichia alive. They scrape the bottom of the vat, and they find the Escherichia that are still alive. And what they did is they then sequenced the genome of the Escherichia that were sensitive, the vast culture sensitive to the virus, and the stuff that was resistant. And they found that there was a small difference in the genome of these two populations. This is a massive selective pressure, right? There's this massive sweep of virus, almost everything dies, a small thing lives. What allowed them to live? Well, it turned out that the bacteria that lived had acquired in their genomes, in the bacterial genomes, had acquired a viral sequence and used that viral sequence to fight off the virus and thus survive the viral infection. Now, this was in 2007, and if you had said in 2007, do bacteria have an immune system? They would say, no, that's the wrong answer. Go back to your AP exam, try to find a different answer. Bacteria do not have an immune system in front. You're confusing prokaryotes and eukaryotes. Well, it turns out bacteria do have an immune system, and no one knew about this until, maybe 2007, 2012, the exact date. It's a little and we want to give credit to. But bacteria have an immune system. When I was a graduate student, starting at MIT in the year 2000, if you would, all my peers, because when you're 22, you know everything, right? You know that. No one went into bacteria. We were in the biology department. What are you going to study? I'm going to go study cancer. I'm going to study immunology. I'm going to study neurobiology. Very few people went off to study prokaryotes because we knew there was nothing new to learn about prokaryotes. How wrong we were. So this is how this system works. The other thing about biologists is you can't be a biologist if you don't like coming up with acronyms. We sometimes do experiments, but we mostly spend our time coming up with acronyms to describe the systems we develop. So CRISPR stands for Clustered Regulated CRISPR, short, no repeats. The acronym CRISPR sounds good. And basically this is the immune system. So an invading virus comes in, but the cell already has in it, cells that are resistant, a scrapbook of viral memories. And it uses the DNA from that scrapbook of viral memories to make an RNA that then cuts the bacteria. So that's how they're resistant to it. They're transcribing an RNA that then cuts the invading DNA, and thus the virus can't replicate. And the great part about this system, so this is kind of how it works with bacteria. The great part about the system, and the great part about biologists, is that, and you saw this wonderfly in Chris's talk, is that you discover a system. And you immediately make that biological discovery, you immediately make it a tool. Like that happens over and over and over in biology, that the discovery of one day is the tool of the next day. So what we can do now, not just in bacteria, but in any system we want, this has been done very notably in human cells and mouse, but in all sorts of model organisms. You just need two components. You need a protein, this blue blob, and you need an RNA, this red blob. And the protein is known as Cas9. It's an endonuclease. An endonuclease means it's a protein that cuts DNA, and it cuts DNA based on whatever this sequence of RNA is. And you can program this RNA sequence to be whatever you want. So as long as you can express an RNA, express a protein in a cell, you can direct a double-strand DNA break wherever you want inside a genome. Now, a double-strand DNA break opens up a lot of possibilities. The simplest possibility is to insert a double-strand, is to cause a double-strand DNA break and then do nothing. Now, this happens with unfortunate regularity in our bodies, and there's actually a nice display right over there about DNA damage in human cells. When you go on a plane and there's ionizing radiation, you're getting double-strand DNA breaks in your genome at some low level. But the cell has a process to repair it. And that process is usually something called non-homologous end joining, NHEJ, where if you get a double-strand DNA break, the cell just kind of shoves it back together. But it shoves it back together in a way where sometimes it shoves it back together perfectly, but oftentimes it shoves it back together in a way that inserts or removes a couple of bases, and that's called an indel, short for insertion or deletion. So when you have an indel, if you're in a region that is coding for a protein, that disrupts the protein. So that is how we're able to that's how we're able to break DNA. Now, what I really like about this is, so this is the CRISPR system, which again was discovered in 2007 and was first applied to mammalian cells only 2 years ago. That's how we take a tool from biology to break DNA. We also have a tool called RNA interference, or RNAi, that's a little bit older. It was discovered in 1998 and first used in mammalian cells in 2001. We now have two ways of breaking this flow of information. We can modify DNA, we can modify RNA, and we can see what happens. So we have multiple modalities. So CRISPR targets DNA, RNAi targets messenger RNA, and when we think about drugs, small molecules that we take, almost always that's targeting protein. Now there's no even remotely simple system to go from the sequence of a protein to designing a small molecule that's going to target it. The programmability of RNAi and CRISPR is easy because it's just based on nucleotides. But drugs are doing this at the level of the protein. So what we do is we use these tools to do functional genomics. And functional genomics is ask the question, well, what are the genes involved in X? And X can be sort of any model, any question that you're interested in. And so all the experiments that we do are basically built on three legs. One of them is the model. The model could be red blood cells grown in a tube. The model could be a mouse with a lung tumor. The model could be cells that grow in the presence of drug. So you need a model of some sort. You need an assay. The assay could be, do the cells keep growing or not? Are the cells, if I give them a drug, are the cells resistant to that drug? The assay could be, if I challenge these cells with the parasite that causes malaria, can that parasite get in? So we combine those two things along with the perturbation. We take those cells and we perturb one gene at a time and ask, well, which gene causes the phenotype of my interest. The vast majority of genes, so we'll go back to that example of malaria for a moment. Well, what are the host factors that affect Plasmodium invading? So Plasmodium, I don't need to harp the fact that malaria is a really horrible disease that is endemic in many parts of the world. And one part of the very complex malaria life cycle is to divide, or is to replicate in red blood cells, or to invade red blood cells. So we ask the question, okay, what are the proteins in a red blood cell that are required for malaria to get in? So we can use RNAi, we can use CRISPR to knock out, to perturb all the genes in a red blood cell, challenge them with malaria and say, hey, which red blood cells no longer were successfully invaded by the parasite? Because those would be great drug targets, and we've actually done that. Another example is drug resistance to cancer therapy. So here's a picture from a clinical trial where this is a man with metastatic melanoma. He is then treated with a drug called vemurafenib, which very dramatically, this is 3 months later, 2 months, 3 months later, very dramatically reduces all the tumors in this man's body. Incredible single agent therapy. However, a couple weeks later, all the tumors come back. So drug resistance has arisen. You give a small molecule that targets the cancer cells, however, there are cancer cells that are resistant to that therapy. So what we did is we took a model of melanoma. We then asked, well, what are the genes that are allowing this resistance to occur? By perturbing all the genes in the genome and finding what allows resistance to occur and what can prevent resistance. So there are many more examples of systems you can take where you can ask, what are the genes involved in this process, and apply CRISPR technology, apply RNAi technology to do functional genomics on it. And that's what I spend my days doing, and that's a lot of fun. But CRISPR technology has also been in the news very recently because it also allows gene editing. So this is the pathway that happens if you sort of do nothing. You break genes. But if you cause a double-stranded DNA break and provide a template, you provide an exogenous piece of DNA, then that allows you to do gene editing. Now we've been doing gene editing in mice for decades. There are many mouse models of cancer, mouse models of autism, mouse models of this, that are all based on modifying the genome of a mouse in some way. And that's totally non-controversial. But of course, the question is, what if we wanted to do this on humans? When would it be okay to actually edit the genes of humans? Now, first, in order to do this as a therapy, you need to be able to take the cells out, modify them, and put them back in. You're going to apply CRISPR technology. If you're going to modify the DNA of a human cell for a therapeutic purpose, you need to be able to take it out and put it back in. So cancer, if we could take out all the cancer cells, we would stop there. We would not want to modify them and put them back in. So cancer is not a disease where specific, directed gene editing is going to do it. But what about a blood disorder? A blood disorder where sickle cell anemia is a great example, right? I mean, that's based on a single base mutation. So you can imagine taking out the stem cells of the patient, modifying it so that they no longer have the sickle cell allele of hemoglobin, and putting it back in. And that would be a complete cure for sickle cell anemia. Likewise, there are people in the world who are completely resistant to HIV because they have a mutation in the gene CCR5 that people think occurred, originally entered the population during the bubonic plague. So there are people around that have still deletions to CCR5, and they are homozygous null for CCR5, and thus they can't get HIV. I have my genome sequenced or profiled at 23andMe, and I apparently can't get norovirus, also known as cruise ship virus. Not that I have any interest in going on a cruise, unfortunately. But I can't do the sugar that gets attached to the surface of cells, I don't make it, and thus the norovirus can't bind to my DNA. Now, I'm not saying that cruise ship virus is something that we really need to rid the world of. Kind of a high-class problem. But in theory, one could do that with gene editing. But the real ethical question comes into, okay, what if, so that's doing somatic cell editing. That's editing, that's editing my blood cells, but that could not get passed on to the next generation. What about germline editing? Whether that's editing sperm, editing eggs, or editing the zygote. very strongly, the scientific community has come out and said this is not something we should be doing for ethical reasons. But let's move past the ethical reasons just for a moment. Why would we not want to do this? I have 3 reasons for why we would not want to do this, even without debating the ethics. First, it's not obvious what disease you would use this for. Okay? There are many diseases that are, when there are single disease-causing genes to avoid, we already know how to avoid them. And we do that through in vitro fertilization, followed by pre-implantation screening. And there's no ethical baggage to that whatsoever. In vitro fertilization is as old as I am. Louise Brown was born in 1978. We've been doing IVF ever since then with no real problem. So if you know that you're a carrier for a specific disease allele, and your wife or your spouse is also a carrier, you can get tested. And you can only implant the embryos that don't carry the allele. There's no reason to do any editing, because we're already doing the IVF and we're already picking it in. Coupled with that, there are many things that we think, oh, it'd be great, we can increase the intelligence of the race, we can have everyone jump higher, blah, blah. Almost any of the sort of more desirable characteristics one would want are an incredible combination of many, many, many genes, no one of which we really understand, and certainly the network of which we don't understand whatsoever. So the idea that we're going to go in and modify, tens, if not hundreds of loci in order to increase your IQ 2 points, that doesn't pass the sniff test. I mean, that's not practical now. I don't know if it'll ever be practical. And then the final part is, the old-fashioned way of making babies is pretty good. And from a population standpoint, we are not going to be reproducing as a species by IVF, followed by gene editing anytime in the near future. We're going to continue to do it the old-fashioned way for a very long time. So I don't think we're going to be overrun with a race of designer babies ever, really. So in closing, I would just like to point out what a great time it is to live in Cambridge and to be in Kendall Square. Kendall Square is the hub of biotechnology on the planet. There are great tech places, there are great pharma places, great academic institutions, obviously. This is the epicenter of it all, and it's a really special place that, on a random Wednesday night, it's Wednesday, right? Yeah, it's Wednesday. On a random Wednesday night, you can come give a talk and dozens of people show up to hear you talk about science. That's kind of not true anywhere else on the planet. So with that, I'll conclude by saying, when the sequence of the human genome came out, Eric Lander, the director of the Broad, was asked to summarize it in 7 words. And he said, the genome, bought the book, hard to read. Had the sequence, but we didn't know what it does, what all these things are doing. And that's really the quest that many of us have been on over the last decade and a half. So we co-opt solutions that are provided by evolution as tools to interrogate gene function, such as RNAi, such as CRISPR. And it's really by learning what genes do that is going to allow us to understand how gene dysfunction causes disease, and then what can we do about it to actually have an impact on human health. So with that, I am done, and it is now time for a small group discussion.
So you guys have had the opportunity to hear a little bit about some basics of synthetic biology, how it's being used, and some of the tools behind it. So now I'd like you guys to group around your tables, people in the back maybe come in and talk to each other about what you've heard tonight. We're going to be passing out some iPads that have access to Twitter, and so we're going to be having a Twitter live stream going on the screen to maybe help guide your discussions or spring up some questions. And so if you guys have any questions that you'd like to ask each other or our presenters, you can add them to Twitter as well. You need to use the hashtag MIT Soapbox. It's on the little triangles on your table. So we can find it on our Twitter stream. And then that way we can continue the conversation as small groups. We're going to do about 15 minutes for the small group conversation. For our online viewers, this is your chance to talk to some scientists through the breakout rooms. And then we'll reconvene together for a larger group discussion with questions for our speakers. Great, thank you.
I know these what interests you about documents? He's talking about most likely. Yeah, he knows. Well, that's why.
So you actually bring where did you go? So it's mostly
Thank you. You guys look so nice in your discussion. Can I just hear your back? I'm going to snap a picture. You continue.
Yeah, no, I think
There's a question. The only way to really do that is
Can you ask if there are any genetic circuits we can I mean, I would do more stuff in another. You know, I really feel like I would like to go very far. But again, like, you gotta set up at least be able to do basic right to implement different sensors to equalize and make it work. I think it's perfect because it's quite simple. This is not sort of one cheaper indicating the sensor more. And it's a cool thing to show someone that many people because talking about sugar gradients, chemical gradients of the chemist, it's pretty hard to extract the sensor. Yes, that's one of the
We have about 2 minutes of the small group discussion left before we meet up as some large groups. We have about 2 minutes until we're going to do large group discussions. So get those final questions in. Tweet your questions here.
That's why it's suggested to keep the sensors and technology as part of this. And so to show yes,
All right, so it sounds like conversation has died down. So now let's open it back up to a large group discussion. If we could actually have John and Christopher come back up to the front so you guys can ask questions that way. And so if you guys have questions in the audience, you can either raise your hand and we'll bring you the microphone so everyone can hear it, or you can tweet it if you prefer to not say your question out loud, and we'll try to alternate between in-audience questions as well as online questions. So I guess to get things started, I have a quick question for you guys. What's the limit on the number of genes that you can add into an organism? Is there a limit?
There's not a limit with respect to the size of the genome. So in fact, people have taken, for bacteria, taken an organism with one whole genome that was about 3 megabases of DNA, 3 million base pairs, and they put a whole second genome inside as well, and it was fine. So we're nowhere near being able to design something of that scale, but you can put in entire genomes worth of DNA.
Do you have any questions from the audience? Looks like we'll go right here.
I have a question for Chris. You said in one of your slides that there was a great many number of sensors that you can use. Is there somewhere in the library where I can go and find a description of sensors, sequence, and how it works so I can get it and use it for myself?
So not yet. So there are a couple of reviews that go over all the different types of sensors, including a few that are also in papers. But this kind of notion where you pull out and have a piece of DNA and that's your sensor and it has very defined input and output, that's actually a relatively new concept in genetic engineering, believe it or not. And so compiling how every group sort of presents their own information in different ways into a single source where you can go in, like a Texas insurance catalog, and pull out what you want. That's an individual. You still need a bit of an expert on.
A good source for that stuff, though, is iGEM and IGEM. It's a synthetic biology group that actually just had a conference in Boston this past weekend where they had teams of high school students come together and sort of try to build synthetic biology groups and assemble all the components. I went to the conference for a day. It was really fun, and their website is fairly well organized in terms of what are the parts of this that one can use. I'm sure. I mean, it's sort of it's crowdsourced. Obviously, Chris's research is not crowdsourced.
Question here, Pat. Yeah, but it's more or less similar to the previous one. I found defining the input and output of the cells are really fascinating, but I'm just wondering how easy it is to do for us. For example, if you get your cell platform, conceptually we can do the design, but can we just alter the DNA sequences and then do a bunch of heading to get what we want, like straight away, or is it like very tricky? You have to tinkering around in your lab, try 100 samples and get one word. Where is it now?
So it depends a bit on what it is you're trying to do with the scales. For a lot of the simpler systems, including the ones that I showed, it's fairly at the point where you can take the prediction off of the computer and you have to do pipetting, but not all the old cloning that you used to have to do with restriction enzymes and DNA. And for the most part, put it in and get a system that, for the most part, works. And you might have to tinker it to improve it, but where you have a pretty good shot. The first one, behaving properly. The more sophisticated and obscure the function you're trying to engineer, the more likely it is to be difficult to go back to that field or have to tinker.
And can we use the platform now?
What's that?
The Celo platform.
Before the end of the year, it's going to be released. Okay.
I just wanted to ask, what would a software engineer do to get synthetic biology? Do you have to go to school for a while, or is there going to be ready to it?
Yeah, it's interesting. Actually, from a software perspective, there's now a dozen companies that's all they do is software for synthetic biology. And many of the people that are at those companies don't have formal training in biology. So there's actually an entire industry, as well as more traditional computer science and electrochemistry departments that are providing the training that you would need in order to do that. So the degree to which you engage the wet genetic engineering in order to start having an impact on synthetic biology has changed quite a bit. So there's a lot of ways you can take what you've learned directly and apply it as opposed to needing a lot of secondary education.
Let's take a question from Twitter. So what do you think of the micropigs released by the BGI to be sold as pets?
Well, so if I understand those right, I mean, they did some very low-level genetic engineering to make a pig that's just quite likely to be smaller than a regular pig by disrupting the growth hormone. You know, it's sort of the genetic engineering that it seems harmless and probably is. You do wonder from a business standpoint, are they going to make a lot of money selling micropigs? I don't know. I mean, I don't have a huge problem with it from an ethical standpoint. I mean, if I had a problem with that, then I'd need to really expand the scope of things on this planet I have a problem with that I've otherwise accepted. But it's the sort of thing I kind of wish there were less of a market for it, where someone just said, "Ooh, that's a cute pig." We have plenty of pets. I mean, there are dogs and cats, you know, straight pets that are not being adopted now. We don't really need to be creating new designer organisms solely because they can fit into a handbag.
This question here. Hi, this is Chris. I was wondering if your sort of sensor activation technology is being used yet, or you could speed optimization and drug discovery. Because I could imagine in principle you could create a cellular system where one of the sensors senses some quality of life pathway, and then you activate would be like a self-control at the end, and could you screw the drug by which kind of states which I also need to engineer in sensors like all the kind of off-target effects that you need optimizing so that you're building that result. We also have local phenotypes and there happens to be all here.
Yeah, absolutely. So in my lab, we have a big contract with Novartis to do research that's similar to that. And then my neighbor at MIT, his name is Ron Weiss, and I tend to work with bacteria. And so we're trying to discover natural products that act as pharmaceuticals using some of the technology that we have. In his case, he works with these circuits in human cells and mammalian cells. And so you can create circuitry, for example, to measure RNA levels. And so you might be looking for a particular pattern of RNA level changes that a drug might induce and have that read out. That's essentially a logic operation in a circuit that you can then screen compounds for something that's a little more sophisticated than morphology. So.
And that's a, up the street in Somerville, there's a poop bank. Which you may have heard about on NPR, because the microbiome is this completely untapped source of diversity, even as a treatment in and of itself. Just finding good probiotics that have already been applied clinically that can be very useful. So then layering discovery in microbiome research with the ability to modify those organisms with synthetic biology approaches really offers up a whole host of human diseases that could be treated this way. That a single small molecule treatment would probably never do it.
Let's take another question from Twitter and then we'll go to the front of the room. So with that question, is synthetic biology applicable to treating autoimmune diseases? Is that a potential application?
No, I thought that was for Chris. Synthetic biology treating autoimmune diseases. What do you think?
It's a little out of my area, so it's hard to talk specifics. Certainly in terms of discovery, so for looking at molecules that might disrupt immune cascades or that's a little bit.
Yeah, I mean, I think, kind of pivoting back to your question, it's clear that a lot of diseases that we associate with increased inflammation, like inflammatory bowel disease, there's a clear microbiome component to that. So to the extent that treatments are going to be sometimes directed at the host cells, the human cells, but many times will be directed at the microbiome. And so whether it's re-engineering bacteria to put into those species, into those patients, or just finding better strains of bacteria to give to people without having to modify them. I think that's definitely going to be part of the treatment option for many inflammatory diseases and autoimmune diseases, which are again oftentimes caused by low inflammation. Yeah.
Hello. All right. So a lot of the things I hear for uses for synthetic biology is related to things like design organisms or pharmaceuticals and such. I was curious about its relationship to the construction industry. So would it make sense that some of these synthetic materials could be used as a way to build better building materials? As an example, I saw someone use small bacteria that was waste product was calcium to continuously build like a regrowing foundation for building the inside.
Yeah, it certainly, and also for alternative routes to concrete, for example, that's one of the most one of the most difficult carbon-polluting resources there is in producing concrete. And so people have been looking at using bacteria for biological processes to do that in a way that's environmentally friendly. There's even a company that is in New Mexico called Industrial Biomining or something like that. They work on, believe it or not, a huge percent of the world's copper comes from bacteria. It's like 10%. So where they're able to leach the copper out of the mine drainage, and then that's the way that it's obtained. And they've been looking at engineering bacteria to go after gold and other rare earths, things of that nature. So thinking beyond kind of the obvious places for biology to have an impact, there are these other areas in traditional chemistry, material science, and architecture, all sorts of things where biology, if we get control of it, has the potential to have an impact. Thank you.
There was recent news about another CRISPR system. What is the implication of having that?
Yeah, so the fun part of biology is that there's a lot of it. And the CRISPR technology, this idea that bacteria can have an immune system, evolution hit on that a really long time ago. So in the last couple billion years, there's evolved a lot of diversity in the way that we can apply CRISPR technology. And while the one that I described was sort of the first one used, that was only a couple years ago, unsurprisingly, people have been looking for similar systems across a whole variety of prokaryotes. And as you mentioned, a new CRISPR system was published 2 days ago. So we're going to continue to see incredible amounts of diversity of what these CRISPR proteins can do. And on one level, that's incredibly exciting because they're going to be any one protein is going to have its limitations. But at the same time, I think even if we only had one of them, there is so much that has been opened up by this, even the last couple of years, that we could never discover another one, and we'd still have decades worth of work in front of us. So it's great that the diversity is there, and there are going to be applications for all these new ones that are discovered. But really the key part was finding that first one, because that just unlocked a whole area of biology that had been closed off until then.
Maybe do one more from the screen. So how about we do, so what do you guys think about a synthetic biocircuit kit for high school students? Do you think synthetic biology is something that should be taught more in schools? I know it's something that I hadn't heard much about until the most recent years.
Yeah, my wife is a high school biology teacher, so definitely answer this one. I think what's great about synthetic biology, especially as a teaching tool, is that it teaches you programming, it teaches you planning, it teaches you circuitry, it teaches you molecular biology. There's so many of the basics of science, not just the basics of biology, but so much of the basics of science that goes into doing synthetic biology. And it's at a level that I think high school students can understand. You don't need to understand every last aspect of transcription and translation in order to say, well, here are the pieces I'm going to put together to educate. And it's the cost of ordering a large piece of DNA that comes off of Chris's software is high now, but that cost continues to drop. And bacteria are something that are very easy to work with in the lab. It's something that high school students can definitely approach in a way that doing mammalian cell culture just wouldn't be feasible in high school. So I think, and it is also, and I'm not in this community, so I will give this community a compliment. It is a community that has been very invested in making their discoveries applicable to a very broad audience. So Chris obviously is one example. Jim Collins is someone who's at BU in the Broad, who's done a lot of work with earlier education with SynBio, because I think so many of the people that get into SynBio, they're not trained as biologists. They come into it from some other direction, so it's clear that it's a sort of a fundamental spark of just doing science that lends itself to doing SynBio.
Believe it or not, there's a lot of high school programs that currently do it, and there's a program that's spun out of MIT called Bio. And so if you're interested in that, they're disseminating, so I think it's dozens of high schools around the country now have synthetic biology programs or serious biotechnology programs, and it's very fun. So it's popular.
Also, if you guys want a chance to try out a little bit of synthetic biology, on October 24th, we're having a group from MIT who developed a product called Amino, which is an at-home tabletop synthetic biology kit, and they're going to be trying it out in the museum on the 24th. So if you guys are around and interested, that's a more of a hands-on opportunity. Do you guys have any final words about synthetic biology you'd like to share?
I'm happy to talk to anybody here if you have any further questions. Love advice on how to get involved.
Yeah, it's a great field. You should get into it.
Wonderful. Let's give our speakers a round of applause. the evaluations. If you guys have your name on them and want to stick around for about 3 minutes, we'll be doing a quick drawing for a set of periodic table playing cards, because you can't have biology without some chemistry, right? So if you guys want to turn those in, we'll do a quick drawing, and hopefully we'll see you next week for our second session on breaking the symbiotic. Thanks, everyone.
Appreciate it. Yeah. Thanks very much. No. Yeah.
Thanks for wondering about that. Thank you.
Thank you for coming.